Good night, everybody. Thanks, Shane, for inviting me. It's my pleasure to be here tonight, talking a little bit about the work we're doing. That has been funded by you guys. So my research, we try to use this new technology, and I'll try to explain a little bit further. That is called high-density electromyography. So what we try to do, we measure the electrical signals of the muscles, and based on that electrical signal of the muscles, we can then open a window and see what's happening with motor neurons. So with motor neurons, we try to measure in humans or in people with MND what's happening with specific type of motor neurons. So we think if we understand better what's happening with specific motor neurons, we can then find better avenues on how to treat them, and should be able also to monitor better changes happening there. So the thing we're trying to measure here is what we call motor neuron excitability. So that pretty much means how excitable they are, how easy it is to use those motor neurons. They can be very hard to use. If you try, your brain tries to send lots of signals and nothing happens with those motor neurons, you say the excitability is low. But if your brain sends signals and very quickly can use those motor neurons, you can say the excitability is high. So I tried to draw this diagram, and yeah, I can use this, yeah. So your brain sends signals, that signals travel all the way to the motor neurons that reside in your spinal cord, and then those translate into signals that your motor neurons discharge, so they fire or they don't fire. That gets to your muscles, and then your muscles will contract and you produce force. In a state of high motor neuron excitability, that's pretty easy thing to do. In a state of low motor neuron excitability, your brain sends the signals and that motor neuron will fire less. So we are very interested in trying to understand what's happening, what happens with motor neuron disease. So what we're trying to do here is a little bit of translational research as well. So there is evidence for a mice model, and you heard a little bit about the transgenic mice models, that the motor neuron excitability that goes up, so motor neurons get very excitable to start with, and before this disease starts to pre-symptomatic levels of the disease. And then after that, then motor neurons start to reverse, excitability starts to reverse, and it starts to get less excitable or harder to recruit or to use those. That actually moved around. So all we had so far was data from animal models. So what you think and why is that important is that changes in motor neuron excitability really seems to be closely related to the degeneration in animal models, but we don't know if that's the same thing that happened in humans. We think, and some people think that because the excitability gets too high at the beginning of the disease, that kind of put a lot of energy demands on the motor neurons, and the motor neurons might get too overloaded with energy demands, and that's why they start to go down. So we think that tracking changes in motor neuron excitability in people with MND has the potential to be a relevant clinical biomarker of disease progression. If you can find that early on, then you might be able to identify people at risk earlier. And also be better to try to track the progression of the disease. Another thing that's important and that we're trying to look at is that not all motor neurons are the same. We have large motor neurons and have smaller motor neurons. So the larger motor neurons are the ones that are able to make big muscle contractions. The small ones, they are the ones that they are not able to make those big contractions, but they last for longer. If you ask for like a long time contraction, the muscle can keep contraction for a long time. And also there is data from animal models that show that disease progression could be different in those types of motor neurons. Like the large motor neurons seems to be the ones to be affected early on, and the smaller ones, they tend to be more resilient, and they seem to last longer. So all the measurements and all the current methods we have to try to look at those things, they don't differentiate between types of motor neurons. We have just an average measure of everything together, so it's very hard to understand if any treatment would be maybe slowing down the progression in one type of the motor neuron, but might not be in the other one. Or even different type of motor neurons, they could be at different stages. One could be going to hyper-excitability, while the other one could be into hypo or lower excitability, and they could be cancel each other, and you're not going to be able to see any result or effect of a drug or a treatment because they're averaging each other out. So what are we doing? So using this high-density MG, and that's pretty much an electrode. So it's It's a tiny electrode. It's a piece of tissue that we have electrodes in. We have 64 electrodes in that. We put on the surface of a muscle. And because you have so many data from different points, you can then put that into an algorithm. And that's non-invasive. It's painless. So you don't have to put needles in anyone. It shouldn't be painful. It's quick and being quiet. You can get a measurement done in 10 minutes in the hospital. And based on that measurement, we can then get the discharge rate, so the behavior, so what's happening, the behavior of all the motor neurons within that muscle, and using the muscle in front of your leg here, the tibialis anterior. And also, with that method, we're able to differentiate between small and large motor neurons. So, again, we place electrodes in front of the muscle, or on top of the muscle. That also gets electrical signal. That electrical signal goes to the computer, and that has an algorithm that's able to kind of detangle all the data. It's quite complicated things. But as you can see here, you can see the red or the warm colors. They are lower, smaller type of motor neurons. The cold colors, they are larger motor neurons. And each color represents a firing. So you can't see here because it's really small, but there are a lot of it's a raster plot, so it has a lot of dashes there. So every time there is a firing, the motor neuron is firing, see one of those points in there. So if that happens more, that means it's firing more. So I'll show you an example of that of a control individual, the individual that doesn't have the disease. So we see lots of motor neurons, so each color is a different motor neurons. So we ask people to make a force, and force goes up, and then force goes down. So in a healthy individual without the disease, you can see that all those motor neurons, they fire continuously. You see you have these traces going without stopping. So those are the lower or the smaller motor neurons. The cold colors are the larger motor neurons. But an example of an individual with motor neuron disease, sometimes what we see is that, yeah, the lower or the smaller motor neurons, they kind of still firing normal, but the larger ones are the ones that are having problems in keep firing. They are not able to maintain the firing rates of them anymore. Even if your brain tries to send some, even if your brain's trying to send them commands to try to keep firing. So that's one of the things we're interested in. We're very interested to try to separate those effects. So what our project is trying to do is try to come up with possible biomarkers of disease progressions that are based on the behavior of the motor neurons and measure them from patients in the hospital. We try to identify changes in specific motor neuron types. We think that would be a great avenue, especially if you need to try to explore new drugs, new treatments. That probably could be a more sensitive biomarker. So sometimes that could be targeting specific motor neurons that are already at risk. And if you just have a measurement that doesn't really take that into consideration, it might be harder to see those changes. So we can then probably try to change and specify better the treatment later. We're looking at the tibialis anterior, which is the muscle in the front of your leg, and the biceps brachialis, which are our muscles. We are testing people at 0, 12, and 24 weeks. And then we try to look at the association between those changes and the clinical test for MNZ. So to date, we have tested already 40 people with MNZ at the Royal Brisbane. We are still conducting testing for the second and the third visit, so that's why it takes a little bit of time because they have to come 3 times, and the last visit is 24 weeks after the first visit, so we're still collecting data. Yeah, I'm happy to address any questions you might have, so thank you very much for your time.